Now, let's continue talking about our nucleotides now in the form of nucleic acids. So now we're moving into polynucleotides, which essentially is the polymer form of the nucleotides we discussed in the last lecture. This is when our single unit nucleotides come together chemically to form a polymer. Now, the way that nucleotides form a polynucleotide, or remember, we also will call this a nucleic acid, which is the name for the polymer form, is that they covalently bond to each other through a phosphodiester linkage. And I want to spend a little bit of time going over what this looks like and distinguishing it from another bond that we have seen, which is a phosphoanhydride bond. So again, these polynucleotides, these nucleic acids, like DNA, RNA, when they strand together the nucleotides, they form a phosphodiester linkage. That is where this phosphate group, remember our phosphate group, uses those O negative bonds. So I'm going to remind you that normally our phosphate group is bound to our nucleotide here, but it has these two negatively charged oxygens that are on it. And it is going to use a negatively charged oxygen to bind to a second nucleotide. And again, you can see that here. Let's look closely at this phosphate group here. And you'll see that this bond is occurring normally on the 5' end of the sugar, the pentose of our nucleotide. But when we link up a nucleotide with a second nucleotide, we're going to use an oxygen on our phosphate to then bond on the 3' end of a second nucleotide. So you can see this bond here is binding to the 3' end of our second nucleotide. Now, again, this is numbered based on the sugar, where this carbon is 1', this carbon is 2', this carbon is 3', this carbon is 4', and our carbon in our CH2OH up here is 5'. So remember, this is where the 1, 2, 3, 4, and 5' are coming from. So this is how that phosphate group is attaching itself from one of our nucleotides to another nucleotide. And notice that when you have a chain of nucleotides, the two ends where we stop adding them together are different looking. This is similar to our amino acids. You'll see that on one end, we have a phosphate group that has not added on to another nucleotide. This is what we call the 5' end. That's because the 5' area of the phosphate is open for attachment. Oppositely, here you can see on our other end, the 3' position of this nucleotide is also open. And again, that's because it is not continuing in growth as a polymer. Because that 3' position is open, we call this the 3' end of any polynucleotide chain. So when we're talking about naming a polynucleotide, we typically are going to be using this nomenclature 5', 3'. So understand what it means, where it's coming from. Now back to our bond description. The phosphodiester bond is occurring when our phosphate is binding between two carbons. So notice that this carbon is attached to an oxygen on our phosphate. That is a phosphodiester bond. And again, carbon, there's a carbon here in the sugar. It's not explicitly shown, but remember the backbone of that sugar has carbons in it. So there is a carbon right here, again, bound to the oxygen. And that is where we get that phosphodiester bond. Now, in the previous lecture, we did talk about other molecules that use nucleotides. And remember, we talked about things like AMP, ADP, ATP. These are all an adenosine backbone that have either 1, 2, or 3 phosphates coming off of them. And I've shown that here. If we just stop our phosphate here, that would be an AMP, an adenosine monophosphate. If we added another phosphate group, stopping it here, we would have ADP, adenosine diphosphate. And of course, if we have 3 total phosphate groups, stopping here, this would be ATP, adenosine triphosphate. Now notice that as we add a phosphate, the phosphates also link together using this oxygen that normally would be linking to our sugar. So when you have an oxygen that links two phosphate groups together, this is called a phosphoanhydrate bond. So again, the difference is when a phosphate is linked to a carbon, as shown here, this is a phosphoester bond. And in particular, when it is linked to 2 carbons, like in our DNA molecule here, it's a phosphodiester bond. And when a phosphate is linked to another phosphate through an oxygen, this is called a phosphoanhydrate bond. So 2 types of bonds utilizing phosphate that we will be seeing a lot when talking about nucleotides. Now, polynucleotides only grow linearly. This is similar to our amino acids. Remember that when we link amino acids together, they only grow in a linear chain. So if you had an amino acid chain, for example, something like this, and so on, these only link linearly, but then they can fold themselves into a 3-dimensional structure. Sugars, we talked about, do not have to be linear. Remember, we talked about glucose potentially having a branched type structure, especially in things like starch. Amino acids, they only will link to each other linearly, but again, we're going to talk about instances in which they can fold into 3-dimensional structures. So we only will ever be able to read our chains of nucleotides in a linear fashion, and similar to amino acids, we can write these using their 1-letter code. So for example, like this means you have an adenosine bound to a thymine, bound to a cytosine, adenosine, thymine, thymine, guanine. This is what this type of 1-letter code chain means. Now, as I just said, we always read these chains from a 5' to 3' end. Again, similar in nature to our amino acids, where we always read from the amino end to the carboxyl end, we have a way to write and read our nucleic acids. So this is usually shown by putting 5' and 3' written next to our one-letter codes. This distinguishes which end is which. Now, what are some differences between a polynucleotide in DNA and RNA? Well, one difference is their stability. The backbone of a DNA is fairly stable. It can last for a long time. For example, DNA that came from mammoths has been isolated. This is obviously hundreds of years old, but because it is in a helix form, DNA is fairly stable. Now, it can be broken down, and its breakdown occurs through enzymes called Dnases. So, Dnases usually exist within living organisms, and they will accelerate the breakdown of DNA. RNA backbones are, in comparison, unstable. So, for example, in water, RNA may last for a few years at tops, but in things like cells, RNA is degraded within the time of a few hours. And similar to DNA, RNA also has an enzyme that accelerates its breakdown and is called RNase.